don't really understand what's happening with my screens right now. So bear with me. Um, I think screen 2 is what I'm gonna want. I think this is what I'm gonna want. Just hang in there. Actually, I think I'm gonna switch it up. Let's see, escape. This is different. They updated, like I said, they updated my computer. And so like it's set up differently than what I'm expecting. Um, excuse me. Don't know how to control this. This is what I want. I don't know how to control this. This is bad. Why is it presenting on this screen? I can't control that. And I can't even move this once I do it. I wonder, drag this to here. And present it? No, it just does. I have no control over that whatsoever. That's awesome. No, I have no control over it. Okay. So I'm not gonna be able to do the things I wanted to. I was hoping I could save like my edits on my slide while I draw on it, but it's not gonna let me draw on my slides, so I can't save those, but that's okay. We'll make it work. I'm just gonna get frustrated probably. I just feel like that's gonna happen. I have no control over that. They changed the settings on this one. They literally I have a new computer, like it's completely new. So I don't have any control over this. This is fine. It's fine. Okay. We'll make it work. All right, let me shut my door really quick and we'll get started. Make sure that this is all recording. We open chat just in case you guys chat with me. Let me make sure I can see it. I can't hear anything. And of course, when I do this, it closed all my volume controls as well. Turn this up just in case. Okay. That's fine. Yeah, okay. The door. Right, sorry about that, guys. I was trying to put together these slides for this review. I'll post the slides. I was hoping that whatever notes I made on the slides, I could save them and upload it with the presentation, but it's not going to let me do that since I have to draw using a different program for it. It's just the way that it's set up. It's fine, right? So anyway, so we're here. We're going to do this. Um, let's see. I don't want black. Let's see. Um, All right, so we're going to do our final review. Basically, what I'm going to do is I'm really only going over the most missed topics from the previous semester. So again, my online section from the summer, which is my best performing section that I've ever had, they got a 68 on my final average. So I took all of the questions that they did the worst on. So anything that they got that had a less than 50% average. I know you're probably like less than the 50% average. Yeah, less than 50% average. I took those questions and then I tried to refine it down to what I thought was manageable in our time limit here. So I don't want to go over more than 2 hours. I can't really handle it much more than 2 hours talking. My throat is killing me, so I'm gonna do the best that I can with this and see if I can make it work. So yeah, I'm here. I'm here. We're gonna do this. It's fine. Right. So I'm just trying to adjust everything. Let me see that you guys are seeing what I'm seeing. Yes, you are. Okay. All right. So okay, let's do this. All right, so I took the major topics, and so I'm not covering everything. So I'll go over these if you guys have any questions about what I'm talking about, or if there's a specific topic that you want me to cover, let me know. Some of the stuff I did cover in the previous semesters, the recordings that I posted. So if you haven't watched the recordings and you're like, hey, can you go over the immune response again? I'm going to say to you, go watch the old recording because it goes over the whole immune response. And plus any other review that I've covered that stuff. I prefer not to have to recover stuff that I covered in previous reviews. So here I'm specifically selecting out questions that they performed poorly on in previous semesters. So that's what I'm going to go over here. So I tried to put these slides together. I literally started putting them together an hour ago, so I apologize for formatting issues, but I did the best I could with this stuff, all right? So we're going to start off. There are 19 slides, but we're going to go into a little bit more depth on these slides as well. And I'll post the slides after the review, as well as this recording, as well as an AI summary. It just auto-generates an AI summary. Take the AI summary with a grain of salt because whatever Zoom is recording and then it transposes it into an AI summary. There could be errors in that. So don't ever go off if it says AI summary. Don't take it as a, direct verbatim. This is what she said. Always like read it with critical thinking and that sort of thing. I just want to make sure it's recording. It is. Okay. All right. Let's get going. So the first thing I'm going to start with is chapter 5, which is the one of the most missed topics on my final exam. So these are specifically topics coming straight from the final exam that were on the previous final exam. I do not guarantee that the exact same questions would appear on here, and I'm not going to cover the questions that were on the exam, right? I'm covering the topic that was discussed in the question, if that makes sense. So here, one of the topics that was missed on the previous final exam for previous sections was the Helminth subgroups. And so keeping in mind that for our eukaryotes, the major groups of the eukaryotes that we have, what is it? Sorry, closed my stuff. Major groups that we have for our eukaryotes, we have the fungi, we have the protozoa, and we have the helminths. Remember, fungi includes molds and yeast. The protozoa are part of protista. So protista is the kingdom that we're talking about with the eukaryotes. Protista includes protozoa and algae. So those are subgroups, subkingdoms within the kingdom protista. And then helminths are in their own kingdom. And these are kind of subgroups within that kingdom. So it's not enough to know that there are flatworms, roundworms, and flukes. We need to know their scientific names. And so if I'm talking to you about, hey, a patient, so we learn in chapter 23 that I learned about several helminth infections in chapter 23. And in chapter 23, I might discuss that, different types of tapeworms like Taenia solium. And if it's a tapeworm, then it's a flatworm, then it's a helminth. And then what are the scientific names for that? So if I say, oh, you have a patient who has tapeworms, which scientific group does it fall into? Even though I might not have said in the lecture that it is Cestoda, then you should be able to recognize in multiple choice that it is Cestoda. So that's what this is talking about here. So we're going to talk about it in more detail, don't worry. And we're going to break it all down a little bit. I say break it down, but we're going to reprise our understanding of those helminth subgroups really quick. Sorry, excuse me. Just trying to be sure I have all my info open up before I get started. It's just opening it up in random windows now. It's cool. Sorry while I drag things across your screen. Okay. I should have just left this open. All right. So Our learning objective for this one, identify helminths as belonging to kingdom Animalia. All helminths are animals. They all belong to kingdom Animalia. And this is going to include, they're all multicellular creatures. All of the helminths, whether they are flatworms, which are categorized as platyhelminths, or roundworms, which are categorized as nematodes, or in the group Nematoda, or if they're subgroups of those, that these guys are all multicellular. All of them are. Now, even like their egg structures might initially start just like ours do as a fertilized egg, potentially if they have sexual reproduction, or just a multiplying cell if they have asexual reproduction. They might start as single cell, but the fully developed sexually reproductive organism is a multicellular organism. The adult version of all helminths are multicellular organisms. And so that's what we're talking about when we talk about the helminths. All of them. The flukes, multicellular. The tapeworms, the cestoda, multicellular. The roundworms, multicellular. All helminths are multicellular. And so we have the helminth category, which the helminth category is the kingdom. This is basically the whole group of all parasitic worms. And then in the helminth category, we have the flatworms and the roundworms, right? The flatworms being called platyhelminthes. Most of us know that one. That's a pretty easy one. Platy, flat, they kind of go with one another. The nematodes, also known as the group Nematoda, these are the roundworms, okay? The platyhelminthes group, or the worms that we would call platyhelminths, the flatworms, fall into two subcategories. The tapeworms, which are flattened and usually segmented, and so they're usually long as well. These are the cestodes. They're called tapeworms. In the group Cestoda, or the organisms, we would call them cestodes, okay? Those are our tapeworms. And then our flukes, these guys, they're also flattened, right? But they're just not usually long. They're usually smaller organisms. They often look like they often look like this, okay? And then they have little eyes or something like that, and they'll have organs inside of them and whatever. So these are the flukes, and these are what we know as the group Trematoda. Flukes are also known as trematodes, okay? Or I also might pronounce as trematodes, recognize it on the exam, right? So trematoda is where the flukes are. And so in chapter 23, we talk about several different types of helminth infections. We do talk about Taenia, for example, and Taenia being our tapeworms. Any tapeworm, it's not just Taenia that we discuss in chapter 23, but any tapeworm would fall into the cestode group. Our flukes includes schistosoma, Clonorchis sinensis, those any flukes at all that we talk about in chapter 23. So if you're sitting here being like, I don't need to know that it's schistosoma, have you watched chapter 23? Have you done chapter 23? Yes, you do need to know that it is schistosoma. What disease does schistosoma cause? Schistosomiasis. It's in chapter 23. And so the schistosomes are known as the blood flukes. How do they get into your body, by the way? They get in through your intact skin. And so they will migrate in through your intact skin. The schistosomes are found in fresh water. And so this isn't all helminths I'm talking about. This is specifically the schistosomes that I'm talking about here. And so this is where, when I'm here talking about helminths and their subgroups, and I'm just talking about, helminths divided into platyhelminthes and nematodes, platyhelminthes divided into cestodes and trematodes, it's not enough to leave it there now at this point in the semester. You need to be thinking, okay, she's mentioned cestodes, that includes the tapeworms, or she's mentioned trematodes, that includes the flukes. Which flukes did I need to know? Which flukes did she mention in chapter 23, where we talk about the diseases that they cause? Well, you need to know the blood flukes, which are the schistosomes, or like the Chinese liver flukes, like Clonorchis sinensis, for example. And so be aware of which, and that's not all of everything that we covered in, obviously, in chapter 23. We talk about all of the different helminth diseases. For the roundworms, For the roundworms, we had a lot that we talked about in chapter 23, and we didn't necessarily go through all of these that are listed over here. We do talk about onchocerca, which is the cause of river blindness. Onchocerca volvulus is the helminth that's associated with river blindness. It's a worm that migrates into the eye and then leads to blindness. We did talk about Necator. We talk about Enterobius vermicularis, which is the cause of pinworms. We talk about trichuria, which is helminth infection transmitted by undercooked meat. We talk about Ascaris lumbricoides, and we talk about more than just what is listed here as well. Trichinella and all of this other stuff that is associated with nematode infections. So when we start discussing the helminths and the subgroups, questions that often appear on the exam, where I might say, your patient comes in with, an itchy anus, and then upon further investigation, you have diagnosed them with Enterobius vermicularis. Which group would this, or which category of pathogen would this organism fall into? I might not just put helminth, I might put nematoda, for example, on the multiple choice. So that's a common type of question that I have on the final exam. So be aware, what does nematoda mean? It means roundworm. And if you can remember that Enterobius vermicularis is a roundworm, as discussed in chapter 23. So this is getting us into the idea of the level of understanding that I'm expecting on the final exam, as well as specific topics that people did perform poorly on in the past on the final exam. Again, I'm telling you that my online class last semester for my summer class, that they got a 68 average. Take it with a grain of salt, y'all. I'm telling you that all of my students got A's and B's in the summer, and their final exam average was a 68. So there's plenty of people that got less than 68, people that got better than 68. They got what they needed to get to keep their grade. You know what I mean? So take it with a grain of salt. Do not panic, okay? Don't read too much into it. They all got A's and B's. So they can get a 68 average and get all A's and B's. Maybe you can too. So keep it in mind. Don't panic. But yes, this is the level of understanding that I expect. So in chapter 23, I discuss Enterobius vermicularis, and I tell you it's a roundworm. You need to know that it is a nematode because it's a roundworm, and that it's a helminth because it's a parasitic worm. Okay. And so be able to categorize based on what we learned in chapter 5, and then applying that to what we discuss in chapter 23, for example. All right. Anybody have any questions about the helminths or the subcategories of helminths? All right, so I'm going to move on to the next thing. I'm trying. I have 4 monitors, and so keeping my mouse on the screen that I need is really, it's trying because I have them stacked too. Okay, so all right, next one. Chapter 7, the viruses and viral structure. So do you remember the parts of viruses? Like, what do you need in a virus and what things are optional in a virus? So remember that viruses, are they living or not? Again, that is kind of still up for argument in science in general. And so what are the parts of a virus? And remember that the basic components of a virus, what all viruses have. All viruses have a genome of some kind and a capsid. Do you guys remember what a capsid is? It's the protective protein coating that covers the genome of a virus. And if I were to try to delve in deeper to the genome, remember that viruses can have any type of genome. If you're a cell, remember viruses are acellular. They are not cells. What's a cell? A membrane-bound structure that is capable of replicating itself without having to use anything from like a host or live inside another cell or something like that, right? They can make a copy, it has everything that it needs to make a copy of itself. A virus does not have everything that it needs to make a copy of itself built within itself. So that's the difference between a cell and a virus. And some viruses don't even have a membrane around them. Some viruses do. Viruses that do have a membrane they stole the membrane from the host cell. And we call that the envelope. Not all viruses are enveloped. Some of them are. Okay. All viruses have a capsid. All viruses have a genome. So viruses can have a double-stranded DNA or single-stranded DNA genome, double-stranded RNA or single-stranded RNA. If they have a single-stranded genome, whether it's DNA or RNA, that single strand can be positive sense, meaning that when you read the genetic code, whatever 3 codons that lead to an amino acid in translation, if we read the 3 codons directly and get an amino acid directly from that, that's the reading frame. That's the positive sense. If we have to convert the 3 codons, A into U, and U into A, and G into C, and C into G, and then read it to get an amino acid, that's the negative sense. So if we have to make a copy of it to read it, it's the negative sense. If we can just read it straight up, that's the positive sense. So just a reminder about that. Okay, so we have like 6 different types of genomes that we can have. Double-stranded DNA, single-strand positive DNA, single-strand negative DNA. That's 3 right there. Or we could have double-stranded RNA, single-strand positive RNA, or single-strand negative RNA. So that's 6 right there. 6 different types of genomes. All cells, that includes archaea, bacteria, and any eukaryote, right? So remember, bacteria and archaea are eukaryotes. They're each their own domain. Domain bacteria, domain archaea, domain eukarya. Okay, those 3 domains, right? Anything that falls in those categories are cells. All cells have DNA genomes, period. Their genomes are made of double-stranded DNA, period. Okay, so just a reminder about that. Obviously, I could go off onto talking about the different types of genomes of them and comparing them and contrasting them, but I'm not going to get into that here. If you need a review on that, please go back and revisit that stuff, right? But anyways, for viruses, they can have DNA, RNA, can be double-stranded or single-stranded, positive or negative sense, right? So there's a lot of different options for the genome type. The capsid, that protective protein coat that surrounds the genome, also includes the spikes. What's a spike on the protein on the genome? What's a spike on a virus? It's the protein on the surface of the virus that helps it unlock the host cell. That is a very watered down version of what it is. So again, spike is also a very general description of it, but we're used to talking about spikes in terms of COVID. The COVID spike protein is the antigen that you test for in your home COVID tests. And so they are surface proteins that basically interact with surface proteins on a host cell that help the virus unlock the host cell and get inside. Okay, so it's typically if you're a naked virus. So if we don't have an envelope, so if we're naked, no envelope, then your spike is usually part of your capsid. If you are enveloped, again, not all viruses are. If you are enveloped, then the spike is on the outside of the envelope. It has to be on the outside, basically, is what I'm saying. Whatever your outside is your outside a capsid or is your outside an envelope? If your outside is an envelope, then your spike is on your envelope. If your outside is a capsid, then your spike is part of your capsid. Okay, so that's the gist of that. All viruses have a genome. All viruses have a capsid. A capsid is not the same thing as a capsule that we see on bacteria. That is a common problem that I'm seeing y'all make, especially when you're discussing viruses and virulence factors and things like that. Don't get them mixed up on the exam. A capsule is basically a polysaccharide and sometimes protein based coating around a cell. Viruses do not have capsules ever, period. End of discussion. Viruses don't have capsules. That's something cells make. Yeah. It prevents phagocytosis and helps with adhesion to surfaces. And in eukaryotes can even make up part of the extracellular matrix. So that's what a capsule is. A capsid is something that only viruses have. Only viruses have a capsid, which is a protein coating that protects and surrounds their genome. Only cells have capsules, and not all cells have capsules. I know all of our bacteria did in our capsule staining thing, but in real life, if we look at every single cell ever, not all cells have capsules, but all viruses have capsids. All right, any questions about this basic viral structure discussion or a capsule or a capsid? Any questions about that stuff before we move on to the next thing? All right. That's how this is going to be. We're going to go slide by slide. I'm going to kind of talk about whatever the thing is about, and maybe deviate a little bit from that and ask you guys if you have questions about each slide as we go on. All righty. Next topic, genetics. Basically, people were not remembering about the leading strand and the lagging strand in the Okazaki fragments. In fact, I feel like on the Unit 2 exam, there was a lot of confusion about the leading strand, the lagging strand, and the kind of purpose of all of that. I have a video of DNA replication that I want to play for you guys really quick. This is in the lecture notes, but I want to play it really quick as a refresher before I start talking about this stuff. So let's see if I can make this work. Maybe it's not going to do it. It's just slow. Okay, I'm gonna bring it up.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units. one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Or that is to imply that I'm telling you, this is the very watered down version of DNA replication here. Right. Okay. So that's the walkthrough of DNA replication and what's really going on with DNA replication. All right. So again, what happens is helicase, or oh no, forgot that I don't have my writing thing on. Actually, I can't write on it. Okay. Okay. Helicase here. Helicase unwinds the DNA and opens it up. As it opens up, DNA polymerase. I know we said DNA polymerase 3. The book says DNA polymerase 3. I'm not going to differentiate. So just DNA polymerase, okay? It can only add on nucleotides to a 3' end. So it can't just open up a DNA. And then just bind on and start adding nucleotides to nothing. It can't do that, okay? What it can do, wish there was a way to just erase those. I'm good. Okay. What it can do is it can create an RNA primer and then come in and build off of that, moving inwards, okay, with DNA. And then it will come back and replace that with DNA. Okay, so this would be heading in. Again, this would be 5 and this would be 3. That this would be the leading strand made continuously, okay, in the 5' to 3' direction. That means the other strand would have to be made as we open it up. We will add in parts. Again, we always have to have an RNA primer that we can build off the DNA from, and we replace that RNA as we go. So to keep that in mind. So then we would seal these together with ligase. These fragments, what are these fragments called? Okazaki fragments. The strand that is made in fragments like this is called the lagging strand. The lagging strand is made discontinuously in fragments called Okazaki fragments that are then sealed together by DNA ligase. Whereas the leading strand is leading, it's just being made continuously. Okay, so that is what's going on with DNA replication. DNA replication. This is not transcription. This is not translation. In fact, on the exam, sorry, I have a cough drop. My throat's hurting pretty bad, but on the exam, people did okay on our transcription and translation questions, but they didn't do great with this replication stuff. So definitely, if you don't remember the difference between replication, transcription, and translation, then that might be something that you would want to go back and revisit. I don't know what's going on with my lighting here. Stop that. Anyways, quick reminder about it. Replication is literally replicating, making a copy of DNA. Transcription is taking a DNA template and making an RNA copy of it. Translation is translating the RNA copy into protein in an amino acid sequence based on the 3 nucleotide codon sequence of the RNA. That's my mini summary of transcription and translation. You guys need me to go into more detail on transcription and translation. I do cover it in the review from the previous semester in that video pretty well. So if you guys want a review of that, that is in that recording from the previous semester. It is posted in your finals week module on Canvas if you need it. Okay. Any questions about replication, transcription, or translation? I know that's a big blanket, but I'm not going to be covering it any further in this review. All right, reach out to me if you end up being confused about it later. That's fine. But be sure you do watch that review first if you have questions about it and see if I cover what you're asking about. If not, the videos that are available in chapter 6 are pretty good at covering this stuff. Actually, let me see really quick. I do think I will really quickly move this over here. Like I said, I got a lot going on. Put this here. Bear with me. There's a point to this, I promise. The internet couldn't be slower. This is unit 2. Let's see if I can drag this over here.
DNA replication begins when the enzyme helicase unwinds a segment of the DNA and opens up the hydrogen bonding between the two complementary strands of DNA. DNA polymerase, the enzyme that synthesizes DNA, can add new nucleotides only to the free 3' end of a nucleotide strand. To provide a free 3' starting point, a short primer is synthesized first. Synthesis of the other strand of DNA is more complex. To provide a free 3' starting point on this strand, several short primers must be added. DNA polymerase then adds nucleotides to the 3' end of the primers. Finally, the enzyme, DNA ligase, forms a bond between the short DNA strands.
It's just another way of looking at this.
During DNA replication, the leading and lagging strands are continuously synthesized.
Okay, so pretty short little video there explaining again DNA replication after we just
DNA
talked about it. Oh my
replication.
gosh, guy. And then we're gonna watch this one. You bear with me, okay?
During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase.
You guys remember this stuff? I know it's been a
The
while.
majority of gene expression is controlled by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
Okay, so that was that one.
During transcription, the DNA...
I think this one was operators, operons. I don't remember. Oh, see, I think this one was translation. I just want to knock out like transcription and translation just really quick. I know this is refreshers. I know this is available for you guys. but it's in the context of what we're talking about and reviewing here, and I just want to be sure it's being covered. Yeah, blah, blah,
The
blah.
genetic information stored in DNA is transcribed into messenger RNA and then translated into protein. Translation begins when messenger RNA binds to the ribosome. The initial transfer RNA occupies the P site on the ribosome. Subsequent transfer RNAs with bound amino acids first enter the ribosome at the A site. The complementary matching of 3 nucleotides on the transfer RNA, called the anticodon, and 3 nucleotides on the messenger RNA, called the codon, ensures the correct sequence of amino acids. The messenger RNA passes along the ribosome in short spurts of 3 nucleotides at a time. As this occurs, the initial transfer RNA is moved to the E site, and its amino acid is transferred to the second amino acid at the P site. At the same time, a new codon is presented at the A site. The initiating transfer RNA, which now no longer carries an amino acid, leaves the E site, and the next transfer RNA with a complementary anticodon enters the A site. Each time a new codon sequence moves into the A site, a new transfer RNA brings in an amino acid. The old transfer RNA paired with the previous codon is passed to the P site and then to the E site as the amino acid it carried is transferred to the growing amino acid chain. As the
So you can see here, this is translation.
Proceeds down the messenger RNA, a stop codon is finally encountered. At this point, the ribosomal complex falls apart and the protein is released into the cell.
Okay. So again, just to get out of this. Okay, so that is translation. Those. Just a little reminder, because I know we just walked through all of it. We just watched a little mini video refreshing our memory, I'm sure, about all of this stuff. So replication is making DNA copies. And you might be thinking, I thought PCR was making DNA copies. Yes, PCR is an in vitro, out of the body version of replication. PCR is the same thing as DNA replication. It's just outside of the body. And instead of helicase unwinding the DNA, we use heat to separate the DNA. That's how PCR works. But PCR is the same as replication otherwise. And we are using usually DNA, single-stranded DNA primers in PCR instead of RNA primers, but otherwise the steps are exactly the same. You know, we're going to be elongating based on nucleic acid sequence. So replication is making DNA copies. Transcription is going from the DNA blueprint to make an RNA copy. Don't just simplify it and say that, oh, it's making mRNA, because how do we make tRNA, which is the transfer RNA that we just saw in our translation? By transcribing DNA. How do we make ribosomal RNA that builds our ribosomes that do the translation process with the EP and A sites that we just saw? Ribosomal RNA is made by transcribing DNA also. So it's not just mRNA that we make through transcription. It's all types of RNA made by transcribing DNA at blueprint. Now, most importantly, maybe for our purposes, most importantly, mRNA is our messenger copy, our working copy of a gene. And so the mRNA and this translation is reading that mRNA and turning it into the protein product, which is a string of amino acids. So that's our little mini review of that. Do you guys have any specific questions about any of that? I know I asked previously, but now that we've kind of revised it, we've revamped it, we've summarized it all a little bit more. Anybody have questions about replication, transcription, or translation?
I have a quick question.
Yes.
So we have replication, transcription, and then translation.
Mhm.
Where of like in those, where does the tRNA like fall in?
Okay, so tRNA is in the translation process. So if let me delete this really quick. So, close. This is not what I meant to do. Dang it. I'm trying to figure this out. Where were we at? Here. Okay. All right, I'm trying to pull up. I think it's W. Yes, it is. Okay. On this. Okay, so in DNA replication. Well, why? Why does it have to be like this? Tell that it's been they changed my computer. It's really thrown me off a lot, guys. Sorry. Okay. I really actually want this. Like, stop fighting me. DNA replication. We are taking our DNA strand and then we are opening it up. And then making one copy in that direction and then other copies in this direction and then sealing them together, right? So that's DNA replication, going from DNA to do one DNA to two DNA. That's basically the gist of it, right? So that's replication. Then transcription. We are taking, in this case, we'll stick with the red for the DNA. Taking that DNA, opening it up, and then making, let's do green, an RNA copy. This will lead to tRNA as a product, ribosomal RNA as a product, and then a messenger RNA, which is a working copy of a gene. And then in translation, we are taking that working copy of the gene, that mRNA, okay, and then Mr. Purple, this will match up to a tRNA. This is the anticodon. This is a codon, right? And then on that anticodon, or on that tRNA, we have a specific amino acid that corresponds to the codon-anticodon matchup, and that all of this is happening inside of a ribosome, which is made of ribosomal RNA. Again, that's translation, that's transcription, and that's DNA replication. And then each of these tRNAs will eventually lead to multiple amino acids being coded for, and they will link together. And those linked together amino acids is what will be the polypeptide, which is eventually going to be the protein after it's been folded properly. And then if we add any cofactors, you guys remember that stuff too. If we need any cofactors, remember organic cofactors are called coenzymes. You guys remember this stuff too. So this is DNA replication, making copies of DNA, transcription, making our RNA products from the DNA code, and then translation, using those RNA products to build a protein made of amino acids and using those as like building blocks or tools or machinery in that process. Does that answer your question, Maria?
Yes, sorry, it was in my notes, but I forgot all about it.
Absolutely fine. And I feel like sometimes just walking, like we see the videos, we have a little bit of a refresher, and now walking through that process with that basic knowledge. It kind of just helps bring it all together. Do you guys have any other questions about this before we move on? That's what I'm here for, right? So I think that's helping bring it all back home. And
I have one
refreshing.
question. For
Yeah.
the DNA replication, the
Mhm.
top strand is going to be the leading one, right? And then the bottom
Yes,
one
in
would
my
be,
image on this, yes. Yeah, the top strand
yeah,
is the
so the
leading one.
bottom one
And
would be the lagging with the
yes, Okazaki
Okazaki
fragments.
fragments. Okay.
Yeah, that'll need to be sealed together by DNA ligase.
And the point of the arrow would be a 3, right? And the other end would be 5?
Exactly. Yes.
Okay.
Yeah.
All right. Thank you.
Yeah, excellent. Anybody else have any other questions they want me to clarify on this?
I have one question.
Yeah.
Are Ozaki fragments only found on the lagging strand?
Yes, that's correct. So because the leading strand, let me make little notes on this just because we're talking about it so that it is clear for you guys. This slide doesn't save in the notes either. So if you guys are looking at this, you can take screenshots or go back and review the recording or whatever. But remember this part here, this blob here would be DNA ligase that will seal the fragments together. These fragments are the Okazaki fragments. And yes, at the end of each of these is the 5 and the 3, or this would be the 5 and the 3. And then the same thing, 5 and 3. And so, yes, and so this top strand here, the leading, which is continuous. So no fragments of any kind, no Okazaki fragments there, only the lagging strand will have Okazaki fragments. Makes sense? You guys have any other questions about this?
So
This is
if
good.
They
Yeah.
answered all my questions.
we go from 5 prime to 3 prime and then 5 prime to 3 prime.
Always 5 prime to 3 prime. Yeah. And so we add on the nucleotides to that 3 prime, and this is where we'll add the next nucleotide and so on and so forth. Yeah. And transcription happens the same direction as well. So this will also be made if we're making RNA in this direction, and this would be 5 to 3. Makes sense? It happens in the same way. It's just using RNA nucleotides instead of DNA nucleotides. And again, refresher on that. What's the difference between DNA and RNA? That deoxyribose versus the ribose, using thymine in DNA or uracil in RNA. And remember that deoxyribose and ribose, they're both 5 carbon sugars. It's just deoxyribose is lacking in oxygen. That's the difference there. And we usually have DNA as double-stranded and RNA is single-stranded. We could probably get more scientific with this, but those are our major differences between those two. Any other questions? All right, good. This is good. Good discussion, guys. Okay, so we're going to move on. All right, so next thing that was, missed on the exam is the microbial nutrition stuff. We didn't talk about this a lot in class in our little reviews, in our Kahoots and in our activities and stuff. But if you watch the lecture, it does cover it at the end of chapter 9. The lighting is bugging me. I need it to stop doing that. It's not in my room, it's the camera that's adjusting the lighting. So that's awesome, but whatever. Anyways, so this is that bacterial growth curve. You guys remember this, the lag phase, the exponential growth phase, also known as the logarithmic. Phase. What does logarithmic mean? It means exponential. It means we are doubling every time. So we go from 1 to 2, 2 becomes 4, 4 becomes 8, 8 becomes 16, 16 becomes 32. And you guys know this because that's how PCR works too. That's what logarithmic or exponential growth is. It's doubling whatever you have every single step, okay? Then we have the stationary phase. This is where growth slows, and essentially our growth rate equals our death rate. So there's no increase in our population. And then we have the actual death phase, where now death outweighs our growth. Okay, so that's what's happening there. So just to bring us back to that lag phase, why is it a lag? It's because when we first introduce our bacteria, so we could say this is time in hours, depends on the microbe. Some microbes are much slower at this than others, and some microbes are faster. Generally speaking, whatever your lag phase is, so in this example, our lag phase is 5 hours. This means that it takes about 5 hours for once you take your loop and you've touched it to your plate or whatever, or your slant, our blacktop slants, right? And we swished it in some broth, or we streaked it on the surface of a new slant or whatever. Once we took it from one place and put it onto new fresh media, that time for that bacteria to adjust and realize there are new nutrients and realize that it can grow and then gather up all of its cellular resources to start making copies of the new cell. That takes time. To copy your DNA and build a new cell takes time. And so that is what the lag phase is. It's the cell recognizing it can make a copy of itself and then starting to build its parts. And we don't actually get a double of ourself until we start reaching the beginning of that exponential growth phase. And as long as we can just keep doubling cells and not have to worry about running out of nutrients or space or start having cells needing to die off from old age or anything like that, you're in your exponential. As long as you're just doubling and doubling and doubling, you're in the exponential. Once you start running out of nutrients, and then cells start dying of old age, and we start having like toxins start to build up, that's when our growth will slow. And then we also start having death of cells for whatever reason. And as long as we're just maintaining that population level in whatever is left in that media, then you're in the stationary phase. But once your death rate starts to outweigh the growth phase, and we start declining in our actual living cell numbers, and that's when you're in the death phase. And so, yeah, so those are the different phases. So there's 4 phases: lag, log or exponential, stationary, and death. Pretty basic concept there. Does anybody have any questions about the growth phase for bacteria there? So this does align with what we see in our disease presentation. So with diseases, which was a chapter 13 thing. Okay, so in chapter 13, the disease progression thing, we have your initial exposure, and then we have the development of your first signs and symptoms, and we start having the actual full-blown infection, level of the disease, and then we start having that recovery phase. Now, those kind of align pretty well with what's going on here. So using a different color here, should have just changed it for this. Let's not use that. Let's use red. Look at chapter 13, the disease progression. We'll do this in red, okay? That incubation phase often aligns with the lag phase. Not always, and it's not 100%, because you can even have some growth starting and still be in incubation phase, but it aligns well with the idea of what's going on with the bacteria. Okay, so oftentimes the lag phase aligns well with our incubation phase, and usually our first signs and symptoms will be happening here. And that is called the prodrome or prodromal phase. Prodrome or prodromal phase, okay? Then once we get into this part up here, which I'll say is 2B or into 3 in general, you're at the acute or infection phase, where you start having the worst of the disease. Like you're maxing in the disease. You don't just have initial signs and symptoms. Like when I got sick with this cold, my first symptom that I had is my throat was hurting a little. And I was like, man, I hope this is just allergies or something like that. And then it was like it built from there, right? I started getting tired. I get that malaise. I get achy. Now my throat really hurts. I've had a lot of sinus drainage. I've had a lot of snot. And I mean, I'm on all the drugs and everything. And like I'm still feeling bad. So and so when I was at my worst, which was probably Friday, that's my acute phase or infection phase. That was me maxing. Now I'm probably down here. I'm starting to have my body is killing off the virus that was causing my illness. I'm getting better. I'm never definitely not 100%, but say I'm about 80, maybe 75 to 80% better. I'm getting there. My throat still hurts a lot. I'm not as tired now. But now in that death phase of the actual virus and my body is recovering, that's the convalescent phase of the illness. I'm convalescent. That means I'm recovering from the disease. Have I recovered completely? No, but I am convalescent. I am getting better. And so that's the disease progression, the infection progression that aligns really well with the bacterial growth curve that is associated with this. And whether you're viral or bacterial or protozoal, the concepts of this still just generally applies to a lot of our pathogens. All right, any questions about any of this stuff? All right, next, aerobic respiration. Yeah. So briefly with aerobic respiration, okay. We have in aerobic respiration, the first step is glycolysis. Okay. And so in glycolysis, we're going from glucose to pyruvate. That's the goal. Do we make other things? Yes. Do you need to know about that? Yes. We also make a little bit of ATP, 2 net ATP per glucose molecule. And we make some NADH, which is just an electron source. So that's there. I'm not going to review that whole thing. Y'all know that that review is up somewhere, right? So that's the gist of glycolysis. The next step, I would consider is the intermediate step. The point of that one is to take pyruvate. I don't like how I wrote that. And turn it into acetyl-CoA. We'll make some carbon dioxide and NADH here too, but it's negligible. That's not the point. The point is to get acetyl-CoA there. Then the third step would be the Krebs cycle. We're going to take all of the electrons off of acetyl-CoA and all those intermediate molecules because we combine acetyl-CoA with oxaloacetate to make citric acid, citrate, okay? And then we're going to steal off electrons off of that. That's what's really happening. What's the whole point of it? To steal electrons. What form are those electrons in? They're in the form of NADH and FADH2. These are coenzymes. Remember those. They're a type of cofactor, right? So these are coenzymes that carry electrons for us. What are they really? In all intents and purposes, they're just electrons for us actively, right? So they're electron carriers carrying around the electrons because the next step is our big daddy step, which is the electron transport chain, where we're going to take those electrons and leftover H pluses. Because remember, this H is really just an electron and an H plus, right? So we're going to take those electrons and H pluses and shuttle the electrons through the electron transport chain, which each of these molecules are called cytochromes. You remember cytochrome C oxidase? This guy here. Okay, so if we're aerobic respirators, okay, it'd be that guy there, okay? So anyways, our cytochromes, we're going to shuttle the electrons through there, which is going to allow us to pump the H pluses out and then collect the H pluses out here. Purple is not the best choice because now it's blending in with the thing. That's okay. The H pluses that are building up out here generate what's called the proton motive. Oh, don't be like this to me. I can do better. Proton motive force, okay? Just want to be able to see it. This is the idea that we're collecting a positive charge on the outside. Relatively speaking, the inside should be comparatively, again, in this immediate vicinity, comparatively negative on the inside. This means that those H pluses are going to want to diffuse. Again, what's diffusion? Movement of molecules from a high concentration to a low concentration because they want to even out. So they're going to move by diffusion through ATP synthase here, and then make ATP as a result of that. So that movement of those protons through there is what drives all of that. So we had electrons and protons in the beginning. We just moved them through some molecules, and that allowed us to make ATP because we still have protons and electrons at the end of all this. So those protons plus those electrons with half an oxygen will lead to making water at the end. And that's our summary of what's happening in the electron transport chain, using electrons and then built up hydrogen gradient in order to drive ATP production. And we make H2O as waste. All right, this whole process of using the proton motive force made by moving electrons through the electron transport chain, this whole process is called chemiosmosis. It's osmosis, which we learned is the movement of water molecules across a membrane, but instead of water, it's chemi. Okay, it's chemicals other than water, but we are using this movement of electrons and then moving protons across instead of moving water across a membrane, basically driving all that because of the charges of electrons and the changing of the shape of all of these cytochromes here and all of that, driving, pumping out those protons. This whole process is chemiosmosis. Okay. Any questions about aerobic respiration or the proton motive force or any of this? All right. Okay, next topic. Now we're in Unit 3. I have quite a few from Unit 3. So the first one from Unit 3 is from Chapter 11, and it's our microbial control methods. Remember, we have our physical methods and we have our chemical methods. And one of the most missed questions on the final exam had to deal with halogens. So this is just a reminder of what halogens are. Halogens are fluorine, bromine, chlorine, and iodine. Any cleaning agent that relies on fluorine, chlorine, bromine, or iodine in its chemical makeup is a halogen. Can we have cleaning chemicals that might have halogens like chlorine and maybe phenols and or maybe surfactants, of course. You can have multiple things going on, and you might have multiple chemicals in a cleaning agent as well. If you have a chlorine atom that's doing actual effects on microbes, then that is a halogen. Halogens, basically what they do, they denature the proteins by those fluorine, bromine, chlorine, and iodine. Basically denature the proteins by, interacting with, we already know this about chlorine and fluoride and bromine, that they're going to have the tendency to be negative. They typically are going to be electron hoggers. are typically pretty electronegative. They steal electrons. General rule, not hard and fast always, but oftentimes. Okay, so they tend to like to hog electrons, and then interacting with proteins and all those like hydrophilic or hydrophobic or amphipathic amino acids that can disrupt how the protein structure basically is formed and then the function of the protein. So whether it's stealing the electron or interacting with free electrons or interrupting hydrogen bonding and stuff like that, the fact that these guys have this negative charge aspect to it can disrupt the protein structure. And that's how this stuff works. And I did not expect you to get that specific with it whenever we were discussing this stuff in the lecture. But if you're wondering and you're wondering how this stuff works, they all work this way. They are all disrupting like electron based interactions in a protein. Okay. So denaturing proteins because of that. This includes anything with chlorine atoms in it, generally speaking, but our biggest ones that we should be aware of are going to be liquid or gaseous chlorine, which is Cl2, hypochlorites, which is anything with OCl. Okay, so it's OCl. So sodium hypochlorite is bleach. So that's an example of a hypochlorite. Hypochlorous acid, which you might see is pretty popular in skincare products now, HOCl. Okay, this is hypochlorous acid. Is related to this stuff, right? Sodium hypochlorite. This is bleach. All right, so they're obviously related. Do you have an H there or do you have an Na? And if you have an H there, you can donate that H as an H plus. And then that OCl exists as a negative. Will ionize like that. Right? You get this idea of this, right? And so this is the idea of what happens whenever these guys ionize in aqueous solution. Hypochlorous acid is a common thing found in your neutrophils. They often use hypochlorous acid in their lysosomes in order to degrade bacteria that they've ingested. So this is pretty useful for degrading bacteria. It also has anti-inflammatory properties and is helpful in skincare, especially if you have skin conditions like eczema and acne and stuff like that. And it's a pretty gentle hypochlorite, as far as that goes. So you can use hypochlorous acid on the skin, buy it in skincare products, and sodium hypochlorite bleach can actually be used on the skin in diluted forms. Don't just do this, okay? Talk to your doctor if you're going to try this. But we can even use bleach baths if you have certain skin conditions and stuff like that. We would recommend diluting it significantly, and then you can sit in a bleach bath for certain skin conditions and skin infections, and even irrigating wounds and stuff like that. You can use these. These are not just disinfectants for surfaces. They can also be used in certain forms as antiseptics. And what's an antiseptic? A disinfectant, but for the skin, right? So remember those terms as well. Sterilizing kills everything. You're not going to have antiseptics that sterilize, because if it kills endospores, it's going to kill you too, okay? So we don't have antiseptics that are sterilizers or used at sterilizing concentrations. And I just said here, our halogens, okay, if you look at this, at all this stuff that's on here, I'm not necessarily going to read everything on here, but halogens are microbicidal and sporicidal, meaning they kill endospores. Halogens do. The certain halogens used at the right concentrations for the right amount of time in the right settings. So again, these are usually disrupted by excess organic matter. Having too much blood or urine or something like that can disrupt their ability to function. But if everything is perfect and we have the high enough concentration and we're using it for a long enough amount of time, you can sterilize. Do we typically use these things in day-to-day at sterilizing levels? No. Are we using bleach typically in a household at a level that sterilizes? Not typically, right? And so if you're using a bleach bath to soak in, you're not using it at sterilizing levels. You're using it at, what I would consider to be antiseptic levels, which is usually, not sterilizing. And so if we're using it on skin, it's antiseptic, or any tissue, living tissue, it's antiseptic. If we're using it on a non-living surface, it's disinfecting. If we're mechanically removing the microbes, like by scrubbing or filtering a microbe, like literally physically removing the microbe from the situation, right? You remember this? That's, what we consider decontamination. Decontamination. So those are our terms there. Disinfection, this would be eliminating, usually killing, destroying, typically destroying the microbes, most of them. And then on non-living surfaces specifically. So that's where a lot of people were missing that distinction when I'm like, tell me about disinfection, describe what define disinfection. Eliminating most microbes on a non-living surface. And we had to make that distinction because if we're on a living surface, it's called antisepsis. So we had to make that distinction in that exam. So just a reminder about that. Be specific, knock out what surface is it on, living or non-living? Does it kill all microbes or just most of them? Or just some of them? And if it kills all microbes, all living anything, then it is sterilizing, period. All right, so any questions about those terms? Any questions about the halogens or anything like that? Anybody want me to go over any other types of chemicals or physical methods or anything like that? Okay, pretty basic stuff, just a little bit of recall issue there, I think. All right, so the next one is the antimicrobial drugs. We have a couple of them from this chapter. This one is our nucleic acid affecting drugs. And then next we'll do the antifungals. Okay. This is should just be refresher stuff. Remember that with our drugs, we had 5 main categories for our antibiotics. We had the ones that affect the cell wall, the ones that affect the membrane. Please keep in mind, all cells have a cell membrane, all. Whether you're bacteria, archaea, or eukaryote, if you are a cell, you have a membrane. Not all cells have cell walls. Bacteria have cell walls that are made up of peptidoglycan. Eukaryotes can have cell walls. We are eukaryotes. Animals do not have cell walls. Okay. Plants do, fungi do, they have cell walls, but Animalia doesn't, okay, no cell walls. Okay, bacteria, peptidoglycan cell walls. All right, so just reminding us about that. So cell wall is different from membrane. Cell wall and cell membrane combined, we call the cell envelope. So that whole outer structure of whatever the cell is, if it has a wall or is just a membrane, that whole outer structure is called the cellular envelope. Includes the membrane and the wall. But the wall and the membrane, they are distinct units. They are not one and the same. They are not parts of one another. They are distinct units. Okay. All right. So antibiotics can affect the cell wall, this cell membrane. They can affect proteins, usually by protein production, usually by affecting the ribosomes. They can affect nucleic acid synthesis, RNA or DNA. Or they can, and these guys usually work by affecting helicase, which is what unwinds the DNA. Usually that's how they're going to work, okay? Or we can affect folic acid synthesis. This is one thing that I didn't talk a lot about in class, and so some people might be like, okay, well, like I said about how bacteria make their own folate, but we need it provided in our diet. So targeting folic acid synthesis is a great target for drugs because we need it provided in our diet. We can't make our own. So if we block bacteria from doing it, it's a good target because we don't make our own. Why do you even need folic acid? It's an important precursor in making nucleic acids and like amino acids. So it's a precursor molecule for those types of molecules. That's why you need it, generally speaking, for any organism. So those are the 5 targets for the antibiotics. Big groups. I'm not covering every single antibiotic here, okay? Big groups in these. For the cell wall guys, we have the beta-lactams. Right, these are drugs that inhibit the cell wall synthesis. This includes psyllins, cephalosporins, and carbapenems. Okay, I'm not going to go right down all of them, but psyllins are our big ones there. Okay, psyllins, cephalosporins, and carbapenems all fall into that beta-lactam group. Other drugs that fall into the cell wall inhibitors include bacitracin and vancomycin and isoniazid, which works a little bit differently, but is for tuberculosis cell walls. What's important in a tuberculosis cell wall? Mycolic acid, right? The wax. And so that's on tuberculosis cell walls. Mycobacterium tuberculosis has mycolic acid wax. And so isoniazid inhibits that. Okay. So it's specific type of cell wall. There's very niche. All right, membrane inhibitors. This is the polymyxins and daptomycin. I'm just gonna put dapto, okay? Daptomycin, okay? All right, our protein synthesis inhibitors. This includes our tetracyclines and related drugs like glycylcycline and doxycycline, the cyclins. A lot of them falling into that tetracycline category. This includes our aminoglycosides, and a lot of macrolides. So these are categories, right? A lot of drugs that end in mycin, not all of them. Mycin, M-Y-C-I-N, okay? Try and fall into those categories. I don't necessarily need you to break it down, like which one is an aminoglycoside or which one is a macrolide. That's not important to me. I do want you to know that macrolides and aminoglycosides often inhibit ribosome function and protein synthesis. And that anything ending in mycin that isn't vancomycin or daptomycin, which are specific outliers, they often fall into this category. So erythromycin, azithromycin, streptomycin all fall into these categories here, okay? All right. Next are nucleic acid guys. These are in a category called quinolones. And specifically, I don't get very involved in the bigger group of quinolones. We really focus on the fluoroquinolones, which are a subgroup of quinolones. All the quinolones affect the nucleic acid synthesis. The fluoroquinolones specifically are the ones that I'm testing you on. So you do need to fluoroquinolones inhibit helicase, which unwinds the DNA. So we can separate the strands and then go through making our nucleic acids. So fluoroquinolones inhibit that. Specific drugs that fall into the fluoroquinolone category that you need to know are the floxacins. All of them. Levofloxacin, ciprofloxacin, anything with floxacin on it falls into this category, okay? Then our folic acid synthesis inhibitors, these are usually our sulfa drugs, sulfa, and trimethoprim. Like I said, the stinky drugs fall into the folic acid guy area. So they are specifically competitive. I need to blow my nose. Inhibitors. Remember what competitive inhibitors are? They bind at the active site of the enzymes. What enzymes am I talking about? Enzymes involved in folic acid synthesis. So if you go from product A to B, and there is enzyme number 1, or B to C, and there's enzyme number 2, drugs can bind at the active site of any of these enzymes. And there's more than one step. There's like 5, 6 steps in this. So to D might be enzyme 3. So we have drugs that can be competitive inhibitors that bind with whatever A usually binds here. A can't bind there with that enzyme anymore because sulfamethoxazole bound there. And so that's what a competitive inhibitor would be. Excuse me while I go blow my nose. I'm going to mute this while I'm blowing my nose so you guys don't have. Okay. Okay. I hope we're good now. Sorry. Okay. So, that's my rundown of the antibiotics. What do antibiotics treat? Only bacteria. Do you treat viruses with antibiotics? No. Do you treat fungi with antibiotics? No. If I'm calling it a hard antibiotic, there's no crossover. There's only one drug in our course. Now, I'm not saying that these drugs can never cross over for anything ever. That is not what I'm saying. But I'm saying for the purpose of our course and what you need to know for our course, let's keep it really simple, okay? The only one that I definitively talk about being a crossover drug is metronidazole. It ends in azole, which I'm going to remind you guys in a little bit, tends to hint that the azoles typically fall into the antifungal category. Can we use metronidazole for fungal infections? Yes, but we don't typically, okay? We usually use metronidazole for amoebas, which are protozoa. Remember, amoebas are a type of protozoa, okay? Amoebas for anaerobic bacterial infections, and sometimes for fungal infections. Sometimes. Typically, metronidazole only works in anaerobic environments. So whether it's an anaerobic amoeba infection, an anaerobic bacterial infection, or an anaerobic fungal infection is more important than what the organism is. So usually anaerobic environments are what are important because oxygen inhibits the chemical from working properly. It literally, oxygen would lead to basically degradation of the drug itself. And the way that it works on inhibiting, it usually affects DNA synthesis as well, similar to this fluoroquinolones. Not the exact thing, but similar, okay? So you don't need to know that part of it, but what you do need to know is that metronidazole is the common treatment for amoeba infections and anaerobic bacteria. I usually test on that. And I don't usually talk about how it's used for fungal infections. Can it be? Yes, but it's very atypical for us to use it for that, okay? Typically, it's these ones, okay? So metronidazole is our weird one. I don't consider it an antibiotic or an antifungal. I typically lump it into the antiprotozoal category because it's most commonly used for antiprotozoal or anaerobic bacteria as well. So you want to call it an antibiotic, you can, because we do use it to treat bacteria, but not only bacteria. So this is a deviant. Okay, this is our deviant. So let's talk about the azoles. We talk about our antifungals. The ones that I test you on are not these ones. Are you going to use these in medicine and actually deal with patients that take these ones down here? Yes, but I'm not testing you on them, okay? The ones that I do test you on are amphotericin B specifically. And you'll notice that amphotericin B is a macrolide. We talked about the macrolides being antibiotics, but a macrolide is a type of chemical structure. That's what it is. And not macrolides are not antibiotics. It's a type of chemical structure. And so many of the drugs that we use to affect ribosome function tend to be macrolides in our antibiotics. But this one, amphotericin B specifically, which on the exam, I never call it a macrolide. I always call it just amphotericin B, not amphomycin B or something else. So recognize the drug names too and don't get them confused. Man, if you guys think I'm being hard on you with this stuff in my class, just wait. Just wait till you get to nursing school and you do pharmacology. because it's not nice. Okay, so amphotericin B. This is an antifungal. So what do you need to know for our antifungals? Just two things. I promise. Now, there are qualifiers to this, right? But generally speaking, the antifungals are just amphotericin B and azoles. Things we exclude from this. We already said one, right? Metronidazole. I usually write in cursive, so I have to think a lot when I write in print. That's why my print is so terrible. My cursive is much nicer, but a lot of people can't read cursive, so I'm not trying to mess with you guys that way. Okay, so metronidazole. And remember, typically the azoles, except for metronidazole, and the other one is sulfamethoxazole. But sulfamethoxazole has a sulfa and a meth. It's like double stinky. What do our stinky guys fall into? Folic acid, antibiotics. Folic acid inhibiting. Okay. Do we have questions about the drugs? Or is there anything else that you guys want me to cover with the drugs? Do you need me to go over antivirals or antihelminthic drugs or anything like that? Or any of the other antibiotics or any of that? Okay. All right. Next is chapter 16. I have 2 slides for chapter 16. So if I'm not covering something here and you want me to cover it, then let me know. Otherwise, I have pretty good in-depth review of the immune response. I don't want to go over the whole immune response here. We have review videos in the past, including my review videos from the previous semester for the final exam, but also other review videos that I have from this semester that I've already posted that cover the immune response. So I don't really want to go over the whole thing here, please. But if there's something specific that you want me to talk about, I will. But bring that back in. We learned about how antigens are molecules that basically our immune system can recognize as foreign. So antigens. Oh no, no. Actually click on this every time. Antigens. Are you kidding me? I clicked on it. Antigens are markers or molecules that can be seen or identified by the immune system. The immune system can see it as foreign. They're usually markers on the surfaces of target cells, but they don't have to be on target cells. Just markers that our immune system can see as foreign. Typically, when we talk about antigens, we're talking about specific markers. So this marker, A, we recognize as a specific surface marker on E. coli. This marker, B, we see it as a specific surface marker on the surface of Salmonella. C, we might see as a specific surface marker on the surface of COVID, whatever. These are just examples, right? And so we have specific antigens that are associated with specific markers on the surface of these specific pathogens. And maybe D is the H1 of flu. And E is the H2 of flu. And F is the N5 of flu. And all of these could be markers that are on flu. And on one flu virus, you might have one H1 and one N5. So you have multiple antigens on the surfaces of pathogens. Obviously, your body can possibly react to 8.6 billion different antigens. So I can't write down examples of all of them, but if we assigned a letter to them, 8.6 billion different letters, right? So those will be all antigens that are specific to, like, each molecule is an antigen. They're specific molecules, each of them. This reason I'm going into this much detail is because a PAMP, so we need to be able to explain what an antigen is versus a PAMP. A PAMP is a pathogen-associated molecular pattern. Now, is a pattern part of a molecule? Yeah. Is a pattern potentially a whole molecule? Yeah, potentially. But it's a pattern that is repeated on multiple pathogens. So, for example, how many bacteria have peptidoglycan? Almost all of them. Almost every single bacteria has peptidoglycan. Which ones don't that we know of off the top of our head? Mycoplasma. Mycoplasma doesn't have peptidoglycan. Okay, we're remembering this? Okay, mycoplasmas do not have peptidoglycan. Okay, example there, right? So all bacteria can have, almost all of them can have peptidoglycan. All of our gram negatives can have lipopolysaccharide, also known as endotoxin. These are not antigens. They're not specific to each pathogen. These are PAMPs. They're molecular patterns that our innate immune system can recognize. This is just a way for our innate system to say, this is definitely not self. I don't know exactly what this is, but it's not me. This doesn't belong here. This isn't a part of me. I don't know exactly what it is, but I know that it's not me. Antigens are what lets your body say, I know exactly what this is. This antigen means that this is E. coli. I see the A antigen, it means that it is E. coli. I see the C antigen, it means that it is COVID. So that's what our antigens are. So they are different because of the specificity of what they're associated with. Okay, so that's the difference there. The other thing that people are getting wrong on the test is epitopes and haptens. So if you have a COVID virus, and here is our COVID, and here's our single-stranded RNA genome in our COVID. Here's our capsid, which is our protein coat that coats the mRNA. And here's our viral envelope, which is the membrane stolen from the host cell, and then it's covered in the COVID spikes, right? That this spike might be the antigen, but the part of the spike that the antibody recognizes right, that round part maybe, the part that it recognizes is called the epitope. The part of the antigen that is recognized by the lymphocyte. In this case, if this is on a B cell, and this is IgD, which is our B cell receptor. So this is how our lymphocyte, our B cell, recognizes the spike antigen by interacting with this part of the spike antigen. That part we call the epitope. Haptens are whole entire antigens. Haptens are tiny antigens. Okay, they're whole antigens that are too small for this situation to happen. So they're too small for this interaction to happen. So in order for that interaction to happen, they need to be bound to something else to make them big enough at first to recognize, to allow all that signaling to happen with our antigen presenting cell and the T cells, the T helper cells. Sorry, throat. Give me a second. All right, so our T helper cells and the T cell receptor recognizing the antigen, and the B cell receptor, IgD, recognizing the antigen and all of that stuff. In order for that stuff to happen, our antigen needs to be a certain size. And if it's too small, then that too smallness, we might not be able to activate any of that stuff in that initial activation step of everything. If we need another molecule to link to it in order to make it big enough to elicit antibody production, we need a carrier. Carrier, there it is. I was like, I know it says it here. Why am I not seeing it? Yes. If we need a carrier to be linked to it to make it big enough so that we can actually activate this whole thing, then that antigen is called a hapten. They happen to be too small to elicit an immune response, okay? So that's the difference between that stuff. This is the breakdown of antigens versus PAMPs, and then subsets of antigens or parts of antigens. Part of the antigen that we interact with is the epitope. Antigens that are too small and need a carrier molecule, they're called haptens. Any questions about this basic stuff? All right, so with antibody, I do have another, I have a third slide on this, which is the antibody. Yeah.
So sorry. Can you
No,
just
you're
go
good.
back again really quick so I could just take a picture of that? That you drew. Thank you.
Yeah, you're good. All right, so next is our FAB and FAC functions. In antibodies, do green. This part here, right? So we see an antibody, and you guys see me draw it all the time with antibodies. I'm like, oh, this part up here is what interacts with the antigen. This is the epitope of the antigen. So this is the spike. This part here that we're actually interacting with is the epitope, right? Right? This part of the antibody, the top of the Y part, like the arms of the Y, is called the FAB. What I consider to be the antibody part, the part that interacts with the antigen, and that's literally what it stands for. FAB stands for antigen binding. The part that actually, the fragment of the antibody that interacts with the antigen is the fragment that is antigen binding, FAB. The fragment that is antigen binding is FAB. All right, FC is the fragment that is constant. Okay. So is it always 100% constant? No. Depending on what your FC region is, the FC dictates. So I know some of us don't like this word. The FC determines the antibody function, the antibody class, and function. So whatever your FC is determines if you are IgD. Actually, let me make a little list over here. IgD, IgM, IgG, IgA, or IgE. There are 5 different antibody classes, and which class you are is determined by your FC. What antigen you bind to is determined by your FAB. Okay, so that's what these fragments are. And so the different classes of antibody that we have, the IgD is the B cell receptor on the surface of the B cell. It is not secreted. It is the surface bound. So whenever we have our B cell, when it is naive, it hasn't ever seen a antigen before, it has IgD on its surface, and then it can interact with whatever that antigen is. It'll take it up. Let's say we're waiting in a lymph node, and our B cell sees our antigen coming through the lymph node. Well, we're checking for the papers for the stuff coming through the lymph node. We'll see this antigen, take it up inside because we picked it up outside. Then we will present it on MHC2. And now we're taking that antigen on there, and then the T cell receptor will recognize the actual antigen on our T helper cell. If it's our T helper, remember I said I wasn't gonna do it, but I'm doing it because I always think about all of it all at once. And I feel like that's the best way to remember all this. Remember, because CD4 recognizes MHC2, et cetera, that comes after that, okay? So this is how naive B cells pick up the antigen. Once they're activated, the first antibody that they will secrete as soluble protein that'll float throughout the body and have antibody function, which we'll talk about next, okay? The first one that you're going to secrete is IgM. It's a pentamer, it looks like a snowflake, and has limited function, but it's what we make first. And then immediately what we will do is start class switching to one of these three.
Does
Yeah.
the plasma cell make what you were just talking about?
The plasma cell, yeah, the plasma cell is the one that secretes the antibody.
Secretes. Yeah,
Yes.
secretes.
Yes,
Okay.
yes. And
Thank you.
yes, so when we're naive, we're just able to use IgD as our B cell receptor to interact with stuff. Then once we're activated, then we'll turn into the plasma cell and it will just secrete IgM first.
And then it goes to
And then
switch to IgG.
to one
Okay.
of these, right? And so we'll get signals from the body during the immune response. We haven't talked about this a lot. We didn't cover it in our class, but what's really going on in the immune response is you're getting a lot of signals, cytokines that are going around. I mean, there are so many, you guys, it's wild, but there are so many cytokines, and depending on what cell signals, what cytokines we're getting sent around will basically determine whether we're switching to IgG, IgA, or IgE, or some combination, more than one than another, or whatever. So depending on what signal you're getting, you might make one or the other. So you'll switch from IgM to usually one of these, okay? So if we're getting signals to switch to IgG, which is the most common one, this is our workhorse. And this is what is basically circulated throughout the blood and the tissues, but it's most commonly transmitted through the tissues, through the blood. And we'll make this one most. If we get signals, we might make IgA, which is found in the mucosa. We call it the secretory antibody. And once we have made IgA, this stuff pretty much stays in the mucosa. And it's just where it will be made is in the mucosa. So in your gut, in your eye, in your lungs, stuff, anywhere there's mucosa, in your mouth, anywhere there's mucosa, this is where this will be. So it'll be in the mucosa itself, as opposed to in the bloodstream. So then the IgE, if we get signaling for that, this one is the antibody that works against parasitic infections. What is a parasitic infection? Protozoa and helminths. And so that's what it's usually involved with, but we also get signaling to make this in the case of allergies, which we talk about in chapter 17, right, with our type 1 hypersensitivities. All right. Any questions about that? I'm going to talk about antibody function next, but any questions about the classes or FAB or FC parts of our antibody? All right, so next is our antibody functions. Yes, so there are 6 functions of antibodies. So once we have, again, just briefly, we have our antigen out here that we picked up on our B cell receptor, which is IgD on the surface of our B cell. How we pick up that antigen. This is our naive B cell. It will then take all of this into the cell, present that same antigen on MHC2. Which the T cell receptor will then see that antigen. The T cell receptor sees the antigen on our T helper cell. The CD4 sees that it's MHC2 that is doing the presenting. Always like this, right? And then we can, oh no. Cross-activate these guys. So this will allow for activation, depending on if our T helper cell is already activated or not. If our T helper is already activated, we can just activate the B cell. If our T helper cell has not been activated yet, we have to activate our T helper cell and make Th2 cells. The Th2 cells can then activate the B cells. Then the B cells become memory, regulator, and effector cells. Our effector B cells are called plasma cells, which will then make secreted antibody. And then this is the function. Remember, first IgM, and then we might class switch to IgG, IgA, or IgE. That takes time though. So first we'll make IgM. So what are those antibodies actually doing? That's what this slide is about, okay? So first, they can coat the surface. Literally just cover the bacteria or the virus or whatever it is and just inhibit it from doing whatever it normally does, binding to the surfaces in the host, the mucosa, whatever it is, just blocking it from being able to bind or interact with the host cells or anything. Okay, so can't do anything, just coat it completely, cover it, right? It's like me smothering you in a blanket. How are you gonna pull your gun out and shoot at me if I have you completely covered and, sticky balls or something like that, right? Oh, geez. All right, next, opsonization, which this is the molecular handle one. Handles for phagocytes to grab onto. So phagocytes, like our neutrophils or our macrophages or our dendritic cells, can come and grab on to antibody once it's stuck on the surface of these guys, and it helps with it eating it up. So that's opsonization, giving molecular handles for our phagocytes. The third one is neutralization. It is different from coating, and it is different from antitoxins. So in coating, we're nondescriptly just covering the surface so that the organism or microbe just can't do things generally. In neutralization, we are specifically blocking docking receptors. Whatever that microbe is using to dock with a specific surface molecule on the host. So the spike protein, blocking the spike protein on viruses from docking with the door keyhole, basically on the surface of cells. So blocking that specific interaction. All right, so that's neutralization. The fourth one is agglutination. This is our sticking together effect. So we're cross-binding with a bunch of microbes and just creating a big old mushed, mashed, stuck together mixture. And so they can't interact with each other or with the host or anything, and we're just globbing them all together. Once they're all stuck together, it's a lot easier for our body to dispose of them and grab onto them and eat them up and digest them and all that. So getting them stuck together very nondescriptly. The fifth one is complement. And specifically, the membrane attack complex of the complement cascade. So remember with complement has three functions. The three functions of complement are to activate the inflammatory pathway, to act as an opson, and leading to opsonization, just like we had with antibody itself. Antibody can be an opson, or complement proteins can be opsons, and opsons are things that the phagocyte grabs on to. So that helps with that eating up process. So complement and antibody can both lead to opsonization, okay? So complement induces inflammation, leads to opsonization, or leads to development of the membrane attack complex. So it's where complement builds holes in the target pathogen, and then it leaks out its contents and dies. It lyses the pathogen. Okay, so it's the membrane attack complex. The three ways you can activate. So complement has 2 threes. What I mean is 3 functions of complement: inflammation, membrane attack complex, and opsonization. Just the 3 functions of complement, but the 3 ways you can activate complement, the classical pathway is going to activate complement through antibody. If antibody bound to antigen, that activates the classical pathway of complement. If lectin bound to mannose sugar on a bacteria, and that activates complement, that's the lectin pathway. If we recognize lipopolysaccharide or other PAMP-like molecules, patterns. If complement recognizes patterns like that, that's the alternative pathway. So LPS is the most common one that activates the alternative pathway. It's the one I usually ask about on exams. But yes, so recognition of patterns by complement is the alternative. Recognition that antibody bound to antigen, classical. Recognition that lectin bound to mannose sugar, lectin pathway. Those are different ways to activate complement and then get those three functions of complement as a result. One of which is the membrane attack complex. So indirectly, antibody binding can lead to activation of the membrane attack complex by complement activation through the classical pathway. So that's number 5. Number 6 is antitoxin function. This is where the actual antibody binds to the antitoxin and renders it unable to do its job. This is how the Tdap vaccine works against the toxins for tetanus or diphtheria or pertussis. So that's how that will work. So literally, antibody plus toxin renders that toxin inert, and so it can't work anymore. It's antitoxin function. So those are the 6 functions of antibody. Do we have any more questions about the immune response? Like I said, I have reviews over them generally. If you need to watch a general review over the function of the immune response, please go back to those. If you guys have any other specific questions about the immune response, I'll be happy to answer them now, because the rest of this, our last slides, are going to be over Unit 4 stuff. Right. You guys can always email me too. Like, this isn't your last chance. So email me if you have questions later, okay? All right, moving on. So now we're in Unit 4 stuff. So this is the Unit 4 stuff that people often missed. I know, guys, I'm not delusional. I know a lot of you guys haven't actually sat down and taken the time to go through the Unit 4 content that much probably yet, but don't skimp on it. Don't wait too long. Review what you need to on Units 1 through 3, but then take the time to at least categorize your pathogens for Unit 4. What I mean is what I considered my C-level work, right? What I mean is that you should, at least for each disease that I talk about here, you should know what the disease is, what the pathogen is specifically that causes the disease, and what type of microbe it is. This is your bread and butter for Unit 4. Now, there's a lot more information than that in some of these diseases, but this is your bread and butter for this. Then you can go back and work on the details on stuff that need details. Disease, what do I mean? I mean like plague. Okay. Pathogen, what do I mean? I mean like Yersinia pestis. Binomial nomenclature, if I'm writing it by hand, capitalize the Y, lowercase the p, underline the whole thing. If you're typing it, italicize the whole thing, right? FYI. All right. Type of microbe, Yersinia pestis is a bacteria. So you want to know, is it a bacteria? Is it a virus? Is it a fungus? Is it a protozoa? Is it a helminth? Or a random outlier, the prion? Everything should fall into one of those categories, and you should know it for all the diseases that are discussed in all of Unit 4. And there's a lot of them. So this is my bread and butter. You should know what the disease is called, what the pathogen is, and what the type of microbe is. So commonly missed on the exam, one of our learning objectives in Unit 4, recall the causative agent of the plague and its defining characteristics, sure. But specifically, people were having issues with remembering the difference between the different types of plague. There are 3 types of plague. They're all caused by Yersinia pestis bacteria, which is a gram-negative rod, okay? They're all caused by Yersinia pestis bacteria. How is Yersinia pestis transmitted? How's the plague transmitted? By fleas. Okay. So fleas transmit it, and that's how it gets transmitted. That's not the only way it can be transmitted, though, because pneumonic plague can be transmitted person to person. Via aerosols. But the bubonic and septicemic plague typically are going to be transmitted by fleas. That's the major vector. That's the major way that we're transmitting the plague, okay? So there are 3 types of plague. Number 1, the pneumonic plague is what it sounds like. It's going to be in the lungs, pneumo, right? So this is respiratory disease version. 2, the bubonic plague. This is infection of the lymph node, leading to buboes. Bubo equals infected and sometimes necrotic lymph nodes. So if the lymph nodes are infected, then it is a bubo. It's bubonic. If we have infection spread throughout the bloodstream, if we have, what is septicemia too? So this is another term that I want to go back to. This is from previous chapters, but I feel like it gets lost a lot in the translation. If we have bacteremia, it means we have bacteria that's just present in the bloodstream. Your blood, as we learn about in chapter 21, should be completely sterile. It should be a sterile environment. There shouldn't be any microbes in it. There's no microbiome for your blood, okay? So if we have bacteria even present in your blood, that's bacteremia. If the bacteria are flourishing, they're multiplying, they're actually causing an infection within the bloodstream and multiplying within the bloodstream. They're not just present there. They're not just transient. They're not just passing through. If they're actually multiplying and dividing and living in the bloodstream, that is septicemia. That's when somebody is septic, is when we have septicemia. So actual flourishing and dividing and living and growing within the bloodstream, that's septicemia. So when we have septicemic plague, we mean Yersinia pestis, Y pestis, is multiplying actively and growing actively in the blood. So that's what that is referring to here. So you can get bitten by a flea and get bacteremia, transient bacteria in the blood that could then grow in the buboes and not really grow in the bloodstream. If it's just present in the buboes, if it got in, you got bit by a flea in your hand while the flea was drinking your blood, a little bit of bacteria got in there, it migrated into your lymph node through the lymph and just stayed there, then that's the bubonic plague. But if it gets transmitted into the blood and flourishes in the blood, now we have septicemic plague. And this is pretty serious. If it doesn't get treated, it's 100% mortality. If you get septicemic plague, 100% mortality rate. It is treatable with antibiotics, but even with treatment, 30 to 50% mortality rate. It's pretty high. So you don't want this to multiply in your blood. It's pretty serious. This is a BSL-3 level pathogen. It's not even BSL level 4, which is like Ebola and stuff like that, where we have to wear the big old suit. BSL-3. So this does mean we won't confine people to a specialized quarantine room because we don't want to risk it moving into the lungs. If you get septicemic or bubonic plague and it moves into the lungs, now you can transmit it person to person by coughing and stuff like that. So this is the 3 different levels of the plague. You probably never thought about it, but they're all caused by the same thing. Flea bite transmitted Yersinia pestis, and then it transformed into these different types of plague, pneumonic, bubonic, or septicemic. And then pneumonic can be transmitted from person to person after that. But usually this is typically considered a zoonotic disease. It's typically found in animals, typically rodents, and they can be fine. They don't have any signs or symptoms. They just are carriers of the disease. And the fleas that bite them transmit it to us. This is found in the United States. By the way, prairie dogs harbor this. Not everywhere in the US, but in the Four Corners is where this is most predominant. But I'm sure you know we have a massive prairie dog population here in Oklahoma. So don't play with them in the wild if you can avoid it, unless Just don't. I'll say that. Okay. So any questions about the plague? Yersinia pestis. It's a gram-negative bacteria transmitted by flea bites. We have the pneumonic, bubonic, and septicemic plague, and know which one is which. All right. All right, next one was antigenic shift and drift. This is associated specifically with flu. This is unique to flu. So flu has antigenic shift and antigenic drift. Antigenic drift, if you're just going with the flow, you're just drifting with nature, okay? You might just have random mutations that pop up. Hey, if your genetic sequence is ACCTGAA, and I'm trying to make a copy of it on the other side, and I'm putting TGG, but here I accidentally put another C. That would be wrong, right? This can lead to a mutation, random mutation, just from misreading the genetic code and stuff like this. This happens all the time. Now, people making new people, having a random mutation like this often can get fixed and replaced before, the person is carrying on this random mutation that gets spread throughout the population rapidly. But if you're making viruses, you're making billions and billions of viruses like every couple of hours. This builds up, and the guys that have traits that help them survive better will keep the traits. The guys that get traits that kill off the virus, it's no big deal to them. If they get a mutation that leads to a deformity in the virus that the virus doesn't survive, it just doesn't survive. We'll make more in the next 10 minutes. It's cool. And they'll have different mutations or better mutations or worse mutations or no mutations or whatever. So this is just random, what we consider spontaneous mutations. Right? And so that can lead to antigenic drift, just normal changes in the surface antigens of the flu from just random mutations. Any viruses can do this. RNA viruses tend to be more susceptible to this than DNA viruses. DNA has just more, just is less likely to have mutations. There's a word for it, and it's just not coming to my head. But yes, DNA viruses tend to be more reliable on keeping their genetic code, whereas RNA viruses are more prone to mutations. Okay. So anyways, flu is a RNA virus. And so it's prone to mutations, just random ones like this. And anything coming from that is antigenic drift, just drifting with nature, just changing according to nature. If you want to make a big shift, antigenic shift happens if you're infected with blue virus and orange virus. Flu has 8 different RNA segments. So if you're infected with 2 different flu viruses, you can get some of the blue and some of the orange and come out with what's basically huge changes suddenly. We didn't just pick up 1 or 2 new genes. We had several genes on each of these segments. We picked up a whole bunch of brand new genes that we didn't have before, completely different versions of what was on those segments, right? So if you have segment 1, 2, 3, 4, whatever, let's just go with 4 for now to make it easy. There's 8. I just don't want to draw them all out. So if you have 1A, 2A, 3A, and 4A in one virus, and the other one has 1B, 2B, 3B, and 4B, and you get infected with both of these, this could lead to a virus that has 1A, 2B, 3A, 4B, or A, who knows, whatever. You can have mixing and matching going on, new gene segments. Well, if 1A had a specific gene for your spike looking like this, and 1B had a specific gene for your spike looking like this, well, your spike might look like this. And if you have another surface molecule, or 2, that looked like this, and this 2 looked like this, well, now you've got a virus that looks like this. This is a new virus to your body. You get what I'm saying? Like your body sees this as a whole new, brand new virus compared to either of these. So this leads to a big shift. And this is because of this gene reassortment. Now, flu is a zoonotic virus. We see it existing naturally in avian, in swine, in horses, in dogs, in all sorts of stuff. And it can cross species sometimes. And so depending on what types of genes you do or don't have, maybe the B virus couldn't infect humans and the A virus could. And A virus caused a very mild disease. And now, because the B has the A on it, it can infect humans. You get what I'm saying here? So this can cause big shifts and allow us now to infect animals that we couldn't infect before. This is the issue and the concern with avian flu, is that right now, while it only infects, birds, and might have gotten more and more severe in birds, all it needs is a shift to jump to humans. And so if it co-infects pigs with the avian flu and human flu at the same time in the pig, and so we just need that ability to cross over. And so it depends. So this is shift. I don't want to get much more complicated than that, but this is when shift happens when we reassort the gene segments in one host, swapping out one or more of the strands of RNA inside of a single host. So the infected host has both subtypes of a virus, leading to a brand new subtype, which is a mix and match of the original two. So you might even call this a C. It's a mix of A and B, but it's a whole new virus to your body. This is usually what leads to influenza pandemics, because there's nothing about this that's like enough for our body to recognize as familiar for us to have enough immunity for it. So we usually see pandemic levels from that. So that's the difference between antigenic shift versus drift. Drift, just drifting with the motions, letting nature change you gradually. Shift, making a big change because we're sorting within a single host. Now, excuse the language here, but this is where I use my little identifier here, because to me, pigs are often the host where this happens, where we'll have a mixture of two different viruses happening. And pigs are poopy animals. So this is where I say shift sounds like shit and it reminds me of pigs. Now, I'm not trying to offend anybody with the language. I'm just saying that's how I remembered it and which one was which, drift versus shift. Okay. Where does the multiple viruses infect the dirty pig that's rolling around in poop all the time? That's the shift. Okay. So that's how I remember that one. If it helps you, I'm happy for you. If it doesn't, don't use it. Okay. Any questions about flu antigenic shift or drift? All right, so I'm going to move on to the next one. Next one is chapter 23, which is our gastrointestinal diseases, and specifically the difference between Hep A, B, and C. We're going to talk about the viruses, we're going to talk about how they're transmitted, and then the vaccines. Okay, hepatitis A. So first things first. First things first. These viruses are not related. They are not related. They cause hepatitis. What is hepatitis? Anytime you have inflammation of the liver. Period. If you have inflamed liver because your gallbladder is blocked up with stones and that led to your liver being inflamed, that's hepatitis. Hepatitis is not always infection. Hepatitis just means inflammation of the liver. That's what it means. So if you have infectious versions of hepatitis, these are some examples in my class that we talked about. There are other types of hepatitis besides A, B, and C. We even had a presentation over type E, for example. So there are plenty of different types of hepatitis. These are the biggies. Okay, so A, B, and C. These are the ones you could get tested on. So hepatitis A, Hep A. This is a non-enveloped single-stranded RNA enterovirus. What's an enterovirus? It gets in through the gut. It is transmitted by the fecal-oral route. Okay. Hepatitis results sometimes, rarely. You can have hepatitis A infection and have very mild disease, maybe flu-like or cold-like symptoms, and not even have any sort of jaundice or anything like that. It's actually not common. Only about 10% of cases will have the jaundice, right? And so this is a fecal-oral transmission, and most people recover from it naturally. But there is a vaccine that is available. Immunization is available. So Hep A, the big take-home message here is that it is fecal-oral and that there is a vaccine. And the disease is typically mild, usually recover. naturally without treatment. That's Hep A. And Hep B is an enveloped DNA virus. It's also a virus that is a DNA virus that has reverse transcriptase. You guys remember what reverse transcriptase is? Reverse transcriptase is what allows you to go from RNA to DNA. Remember, normally we use DNA as our blueprint, and we make an RNA copy, and then we translate that into protein, right? So transcription is going from DNA to RNA. If you go from RNA and make DNA out of it, this is reverse transcription. Reverse transcriptase is the enzyme that allows this to happen. We do not have this naturally in our bodies. Normally, your body cannot make DNA from RNA. And usually reverse transcriptases have specific requirements on how they work and when and where and why and everything like that. So this can be tricky to just blanket also. So anyways, hepatitis B is a DNA virus that has reverse transcriptase, because what it does is it takes its virus DNA genome, it transcribes it, normal transcribing, makes RNA copy of it that we will then translate into protein, but we can also reverse transcribe it to make more DNA, and that's how it makes copies of its DNA. It does not replicate its DNA from DNA to DNA. It goes DNA to RNA and then makes DNA out of the RNA. Don't ask me why. It just does. Okay, so that's how Hep B works. It overcomplicated things. Okay, so anyways, it's relevant though, because anything that's able to do this, RNA to DNA stuff, often can also incorporate this DNA that it reverse transcribed into your DNA, which Hep B does. So Hep B takes this RNA reverse transcribed to DNA, and then inserts that DNA copy into your genome, and that can lead to cancer. So liver cancer can result from Hep B. And often, once you have Hep B, you have it forever. There's no treatment for Hep B. Hep B is forever. So this is why. It incorporates into your genome after that. So it can lead to cancer. Okay. So this is going to be bloodborne, even small amounts of blood. Small amounts of blood. So we can have this from needle exchanges, but we can also have it from sex. Sex and blood transmission here. Okay. Even a little bit of blood exchange can lead to Hep B. There is no treatment. There is a vaccine, though. Vaccine to prevent, but no treatment. And it can lead to cancer. Hep B, not to be trifled with. It's bad. It's a bad deal. Okay. All right. Next, we have Hep C. Okay. Hep C is really, as far as the signs and symptoms, very similar, but the difference here is it takes more blood. It needs true blood transmission, true blood exchange. So we're talking about sharing needles, typically intravenous drug use. Or accidental needle sticks. So that could be blood exchange as well. So that's usually what's going to be associated with. There's no vaccine, but there are effective antiviral treatments. This can lead to cancer. It can also incorporate into, this is, oh, so I do want to say this is an RNA virus. And it can lead to cancer and stuff like that in the liver as well. But when we get to that level of stuff, it's usually dealing more with damage done by the viral infection. So anyways, this is treatable if you are in earlier stages of Hep C. So the earlier you're diagnosed with Hep C, the more likely we can treat it. But it is treatable with antivirals. Okay, so there's the differences in these. Do we have questions about hepatitis? Okay. So these are the differences. So if I asked you to compare and contrast the transmission of A and B, then you would tell me A is fecal oral transmission, B is transmitted through blood. And you could even just leave it at that. You could even just say even small amounts of blood, like through sex and stuff like that. All right, that's the level of understanding I'd be expecting with that. Any questions about it? We'll go on to our last thing. Chapter 24. This is a very niche one. This is a very specific one. So chapter 24, it covers the genitourinary diseases. So basic ones that it covers, UTIs, which is most commonly going to be caused by E. coli bacteria from your own booty, typically. It also talks a little bit about prostatitis, which again, you can get a UTI and then it can move into the prostate, through the ductwork in the male anatomy. And it's usually caused by the same bacteria that we see associated with UTIs, often E. coli. In males, it tends to be from anal sex, unprotected anal sex, leading to bacteria introduction into the urethra, FYI. Also want to bring up, this is talked about in chapter 24, but I did want to bring up because I don't want to get missed, but the idea that people talk a lot about in society about the big topic now being circumcision. So I did want to bring up about this, that there shouldn't be, if you're properly performing hygiene and washing everything in your nooks and crannies and everything, there shouldn't be an issue with hygiene associated with being circumcised or not. But where the issue comes from is that if you are uncircumcised because of the folds and because of the moist area and everything like that in that environment, is that circumcised males tend to be less susceptible to STIs versus uncircumcised males, because being uncircumcised actually introduces a different biome that actually leads to more inflammation, which does make you more susceptible to STIs. So that is a real thing. It's not dealing with hygiene. It just deals with the natural biome associated with being circumcised and uncircumcised and the inflammation associated with being uncircumcised. My throat is hurting. We're making it though. I didn't want to leave that out because we didn't talk about it in class. It is in the lecture videos. It's FYI. So anyways, moving on. And we also talked about leptospirosis. We see leptospirosis most commonly in animals and really most commonly in dogs. It is transmitted via urine. It is a bacterial infection, and it can lead to pretty significant disease and leading to inflammatory diseases as well. And it can be significant as well. It's going to be pretty deadly if it's left alone, and it's kind of harder to diagnose. It's not as common. It is, there are vaccines that are available for lepto, so definitely vaccinate your pets. And prevent that, because it's spread commonly in wildlife to pets, and then it's spread through urine after that. So, and people can get it from their pets and from wildlife. So be aware of that. Bacterial disease. The other ones that were all here were almost entirely all STIs. What is an STI? A sexually transmitted infection. Why don't we call it an STD anymore? Because you can harbor the pathogen and have an infection without presenting with disease. Remember, disease is deviation from health. And it is not uncommon to see people with maybe gonorrhea or chlamydia, or very commonly HPV and even herpes and stuff like that. You can still have the infection and not have disease. And you're still contagious if you don't have disease. You're still contagious if you don't have disease. You're still contagious if you don't have disease. If you have infection, you're contagious. So that's why the language changed. If you're too young to have been around for that, it used to be STDs. Now it's STIs for a reason, because we're trying to educate people. You're still contagious even if you don't have disease. So get tested. That's the only way to know if you have it or not. So this is where we talked about common sexually transmitted infections, ones that were discussed. Oh yeah, we also talked about things like vaginitis and vaginosis. Just briefly, it's not STI related. Vaginitis can be STI related. If you have vaginitis that is associated with yeast infections, then that's not STI related. But if you have vaginitis, inflammation of the vagina, that's associated with Trichomonas vaginalis, which we call trick, which is a protozoa infection, that is an STI. So that can lead to that. Vaginosis is usually because of overgrowth of certain bacteria in the vaginal canal. Vaginosis, overgrowth of the bacteria without vaginitis, inflammation of the vagina. Can you have inflammation of the pelvic organs though? Yes. So you can have things like salpingitis and stuff like that develop even without the vagina being inflamed. So be aware of that. Salpingitis, by the way, is inflammation of the fallopian tubes. Other STIs that were discussed in this chapter, obviously chlamydia, which is caused by the bacteria, Chlamydia trachomatis, the same species of bacteria that was associated with trachoma, which is the leading infectious cause of blindness in the world. But different serotypes usually are associated with that. They're not the same exact bacteria, different subspecies of it associated with each one. I'm not saying that they can't both lead to one or the other, but it's just less commonly seen that way. So chlamydia. We also have gonorrhea, which is a bacterial infection caused by Neisseria gonorrhoeae, which is gram-negative coccus. Neisseria species in general, gram-negative diplococci. Neisseria meningititis was one of the causes of meningitis, and then Neisseria gonorrhea is, obviously the cause of gonorrhea. So there's that one. We also talked about herpes, which is HSV1 or 2. HSV1 or 2, both of them can be oral or genital. Most commonly, HSV1 is going to be oral and HSV2 is genital, but they can go either way, guys. They can go either way. And so herpes viruses are extremely, extremely common, especially HSV1 with oral herpes. If you get canker sores, that's herpes, FYI. So herpes is a viral infection treated with a version of acyclovir called valcyclovir or Valtrex. It can keep the outbreaks under control. But even if you don't have disease outbreaks, you can still be infectious. It's less common with herpes. If you're not having outbreaks, you're really unlikely to be transmitting the disease, FYI. So that can be controlled that way. Herpes, so HSV, we also have HPV, human papillomavirus, which leads to genital warts, as well as cervical cancer. Different HPVs cause different things. There's HPVs, there's like several different HPVs. I want to say hundreds of different HPVs. Some of them are associated with warts that are just on your skin, like your hands, plantar warts on your feet, all sorts of those. Any wart is caused by an HPV. Some of them are genital warts. That's another type of HPV, or certain types of HPV associated with genital warts, certain types of HPV with body warts or foot warts or anything like that. And then some species of HPV virus that are only associated with cervical cancer. Usually the ones that cause cancer are not the ones that are associated with warts, FYI. And usually people will have HPV and have no signs or symptoms at all. Most people will eventually clear the viruses from their body naturally and not have any effects, but this is why pap smears are so important for screening, because it's really the only way we have to detect HPV before it becomes symptomatic cancer. And once it's symptomatic, there's usually not much we can do for you. So it's been the #1 way of preventing cervical cancer, getting pap smears. So if you haven't been getting your pap smears, please do. It's estimated that over 90% of all adult sexually active people in the world have had HPV at some point. So over 90%, we think it's closer to 95 to 99% of adults. Usually clear it, but you probably had it if you've been sexually active. There's ways to avoid that, of course, with monogamy and single partners or, abstaining or whatever, but I'm just saying, okay? HPV, obviously viral. And the other ones. So this obviously is chancroid. It also talks about molluscum contagiosum, which is usually associated with skin disease, but is associated with sex work as well, and other stuff too. I'm not listing everything here, right? Syphilis, which is bacterial. There's 3 different stages of syphilis. Depending on what phase of syphilis you're in, primary, secondary, or tertiary. There's different symptoms associated with each of them. With primary syphilis, we usually see that associated with the chancre, which is what it's called. It's called a chancre, not a canker. That is a different thing, okay? A chancre. and is a site where the bacteria got into the body. And so it's a lesion. It's usually a small, hard bump, often painless. It looks bad, but it's usually painless. And that's where the bacteria got into the body. This can be on the genitals, around the genitals, in the anus, around the anus, in the mouth, on the face, anywhere that it got in. Okay. This is that primary syphilis. Secondary syphilis is most commonly associated with like feeling unwell, swollen lymph nodes, and characteristic rash. I can spell it, I promise. So this is the most characteristic aspect of it. Swollen lymph nodes and the rash, often on the palms, the soles of the feet, or the back. It's like a brownish rash. And tertiary syphilis is where we start seeing neurosyphilis, dementia, and development of gummas. And eventually leading to death eventually after the tertiary stage. A gumma is essentially like tumor-like formations as a result of the spread of the bacteria. Tumor-like, and I really mean like it's more like eroding away at the tissue. So this is where we see the deformities in the face and the musculature and the skin tissue as a result of the presence of syphilis. It can be very disfiguring. So those are the gummas. And then neurosyphilis leading to dementia, and it can affect the pupils and ability to see and lead to blindness and stuff like that. Eventually death if it's untreated. Very easily treatable syphilis with penicillin, usually intramuscular injection of penicillin G. So it's very treatable with antibiotics. I could bring up the thing, just make sure I'm covering everything, because I know that people are, maybe doing our best at studying these later chapters. Covered this. Okay, we'll get to chancroid herpes. Okay. And it talks a little bit about group B strep transmission to babies. So often Streptococcus galactiae or agalactiae, excuse me, Streptococcus agalactiae, which we will test pregnant women for in their vagina prior to birth. And then if they have the presence of that Streptococcus bacteria, then we will treat them with antibiotics to prevent it from being spread to the baby, because it can often lead to deadly meningitis and birth defects and stuff like that associated with infection from that streptococcal bacteria. That was talked about in this as well. That's just part of the normal biome, though. That's not an STI. So chancroid, which I've left for last, it's a weird random one-off one that we discuss in this. Chancroid, it also is associated with development of a chancre, okay? In this case, the chancre is painful. Well, it can be painless. It just depends. Right. Painful in men, often unnoticed in women. But you guys, if you saw the pictures online, I don't know how it could be unnoticed. Okay, because it's a large, soft. It has exudate. We'll just leave it at that. Okay, it's very weepy sort of chancre wound. Okay, and so I don't know how it would go unnoticed. But anyways, this is a sexually transmitted infection caused by a gram-negative bacteria, Haemophilus ducreyi. So this is, again, if we're looking at our thing, what's the disease? What's the pathogen? And what type of microbe is it? The disease is chancroid, characterized by that soft chancre. Okay. Pathogen is Haemophilus ducreyi. Now, on my short answer questions, I've already told you there will be short answer questions. I'm not going to make you write an organism name that's ridiculous like this. Okay. I won't. Yeah, I don't want to see you try. I don't want to see you try to spell it. Okay. So FYI, that's not going to be what I'm going to ask in the short answer questions. I might say what type of disease is Haemophilus ducreyi associated with, and then you'd have to recall that it was chancroid, for example. Okay. I'm not saying that's one of the questions. I'm just saying be prepared for stuff like that. What type of pathogen is Haemophilus ducreyi? It's a bacteria. So those are our basics. Anyways, it's a STI. Okay. Commonly missed question on the exam, because it's one of those one-off ones. I just feel like people kind of, they don't know it. We don't see it a lot in the US, and then they're just not as familiar with it. Okay. That's it. Does anybody have any other questions you'd like to ask me before I call it a day? If that is it, Remember when you guys are studying, email me if you do have any questions. And remember that there are other reviews from earlier this semester that you guys can go back and look at, weekly reviews. I will have my weekly review on Thursday at 4:45, like I always do. So that'll be my last like review that I'm giving with slides. That will be over chapters 23 and 24. So yes, I did record this and this will be posted to the announcements as well as to that finals week module. And so you guys will be able to find this there. I will post the slides as well. So you guys will have those for reference of the slides, the recording, and if it makes an AI summary, I'll make a file of that as well. So you guys have all that for reference. And I will post this in a separate link on that finals week page. And it'll just be like, review from this semester or whatever. So this is my last actual review over finals. Otherwise, you have my recordings from previous semesters. And then, yeah. Yes, let me tell you, I'm not feeling great. So that's why I'm not doing a review on Thursday, except for my weekly one. So you don't miss out on extra stuff about 23 and 24 that I have lined up. Yeah, if that's it, I'm gonna let you guys go. I'm gonna post this recording and post these resources for you guys. Study up, email me if you have questions. Go visit Sydney if you haven't already. Get your course evals done, get your extra credits on, knock out everything that's left. Remember, I don't accept anything late after the exam, nothing. So nothing will be accepted once you start your exam. So get all that stuff in before the exam, and I'll see you either on Monday or Tuesday. Good luck, guys. You got this. You've got this. It's just one last little hurdle. All right, that's it. Thank you guys. I'll see you later.